Hello, my name is Blake Bartlett, and the name of my project is Computer Modeling of Aerosol Particle Transport through Lung Mucosa. So starting off at the very beginning, lung diseases are some of the most prevalent in the world and account for as many as 4 of the most common causes of death worldwide. And these are both pathogenic diseases, which include things like tuberculosis or COVID-19, but also lifelong conditions, things like COPD or cystic fibrosis. And these two types of diseases are going to be cured through different means. A pathogenic disease might be cured through an antibiotic or a vaccine of some sort. Meanwhile, things like COPD are going to require more of a lifelong treatment, a therapy perhaps. But regardless, both of these types of diseases can be treated, have the potential to be treated through the use of an aerosol. Now, these diseases are located pretty much directly within the lungs, and so if we were able to deliver a drug through the airways directly to that diseased tissue, we would be able to maximize the amount of drug located at the diseased area and kind of minimize some of these side effects that are felt when a drug is delivered generally throughout the body through the bloodstream. So there are phenomenal systems in place within the biological lung that prevent this from being kind of a straightforward concept of just having someone breathe in a drug and it helping them. And that is namely the mucociliary clearance mechanism. The lung is coated on its interior with a protective mucus layer that is pushed upwards towards the throat by a layer of cilia. Now, the same mucosal tissue is also present in the nasal passages, where in the upper nasal passages it goes down to the throat, and in the mid-nasal passages it goes down out your nostrils, but we're going to focus mostly on the mucociliary clearance mechanism as it occurs in the lower lungs going upwards towards the throat. And any sort of particles that we inhale in our day-to-day lives, whether it be something that would cause a disease or something that is totally benign, they will impact on this layer, stick to it, and most often will be cleared to the throat, where we would either swallow it and it'd be neutralized by stomach acid, or we would cough that mucus up, removing it from our bodies entirely. However, in the case of someone that's diseased, we might want to utilize this exact same route to deliver a helpful drug. And that's where the modeling part comes into play, because we need to know what sort of characteristics a delivered nanoparticle would need to have in order to diffuse through this complex biomaterial, which is the mucus. Mucus is mostly water, but it's given this very, very viscous, in fact, non-Newtonian character by a network of glycoproteins, and these are called mucins. So there are several hindrances to particles that try to diffuse through this layer, as we will get into a little bit later. Looking here, this is a simplified version of our simulation domain. So it's a two-dimensional representation of this mucus layer, very, very similar to what we see in this figure right here. So the left simulates going lower into the airway, so deeper into the lungs. And the right, or the upper airways, is getting closer and closer towards the throat. And that is shown here in the velocity field, where, as expected in the biological case, the mucus, because of its viscosity, moves as a sheet. It has a constant velocity throughout, and it's cleared from the lower airways up to the upper airways. Now, looking at some of the differential equations that we use to govern the system, it's a complex set of mathematics that goes into physically describing this system. But I would like to point out one in specific right here, our effective diffusivity equation. And that is because we have two components there that give us how this particle is able to diffuse through the mucus, if at all. And that is, one, its hydrodynamic term, which is the actual fluid forces that are acting on a particle as it attempts to move through the mucus. And then also a steric hindrance term, and that is where that network of glycoproteins that the particle could physically bump into is going to come into play. How is it going to be able to navigate this network and eventually reach the tissue below? Now, looking at some of our results, and as you can see with the animation going right here, it doesn't really work when I zoom in, so it's going to have to stay zoomed out a little bit here. We can see that as a particle is administered to this sort of dosage site that you see right here, it will eventually kind of diffuse into the mucus and hopefully at some point reach the tissue below. Now, depending on how thick that mucus is, it's going to change when that particle is able to reach the tissue and in what concentration. So thinner mucus is present lower in the lungs, closer to the alveolar regions. And then, due to the presence of these goblet cells that you see right here, mucus is constantly secreted throughout the tracheobronchial tree, and the mucus layer is much, much thicker closer to the trachea. So these three different simulations you see right here could represent, say, alveolar tissue versus kind of mid-lung versus like tracheal tissue. So differences in mucus layers depending on your physical location in the lung. And some of the results that you see right here, this is for a very thin mucus layer right here and a particle that's only 20 nanometers in radius, just to get you a visual of how penetration can occur here. For a very thin mucus layer, you get a high amount of penetration relatively close to the dosage site. And that can be seen in the converse with the particles that are seen in the second plot right here. So this is the same data as the first plot, but it's on a logarithmic scale, so you can clearly see the differences that different mucus depths will have on the penetration of these particles. There's still going to be a local maximum in the amount of dosage that reaches the tissue, but it's going to be smaller in magnitude and it's going to be further down the domain than for a thinner mucus layer. So in summary, smaller particles are able to diffuse across this mucus faster than larger ones. And thinner mucus is, of course, crossed faster and at higher concentrations than thicker mucus. So at some point upstream of our dosage site, there is a maximum delivered dosage. And that's a very crucial finding right here because it can be used in the development of these disease treatments. We need to know where can we maximize the delivery of this drug to get the maximum therapeutic benefit in an individual.